Welcome to the GobyWeb video training series. In this tutorial, we will look at how to visualize output files from GobyWeb using the Integrative Genomics Viewer IGV. Firstly, we will look at visualizing alignments in the Goby format in IGV, and then we will look at visualizing VCF files outputted by GobyWeb. First, we will look at alignments. We'll assume that the alignment analysis has been carried out in GobyWeb. For more information, please look at the tutorial on alignment. Secondly, we assume that all the associated alignment files have been downloaded. For more information on this, look at the tutorial online on how to download the alignments. In this tutorial, we are using a B-cell lymphoblastocell cell line RNA-seq sample that was aligned using the star aligner, which carries out spliced read alignments. I've already preloaded IGV in the background. For more information on that, look at the documentation on the IGV website. Our sample is human, so we upload the human genome tracks, and preloaded comes the RefSeq gene track. We want to load the alignment files to IGV. We've already downloaded the Goby alignment files. Here they are for this sample. Here I'm going to upload the Goby alignment file, which ends in the extension .entries. So we load this file, and this uploads a GobyWeb alignment onto IGV. We have up here the coverage track, and below it we have the read alignment, and you can see the different reads that align to this gene LAD1. Here you see this is a star alignment, so you see the spliced reads here demarcated with these blue lines. So you can see that you can upload a Goby alignment directly to IGV. The GobyWeb alignment plugin also outputs the bed file as well as WIG file. Here I've indexed these files using IGV tools to create a binary format of the WIG file, TDF. This is for speed in loading, as well as an index file for the bed file. You can also upload those directly to IGV. So here, upload those. And here's the bed track down here. And then similarly here we have the auto scale. We have the WIG track for this alignment. Put that next to the coverage track, and you see that's the WIG file produced by for this alignment. Now we'll change tracks a little bit and look at output from group comparisons. We want to see how to visualize VCF files outputted by GobyWeb. In this case, we're going to demonstrate the methylation VCF file as well as IGV file outputted when you compare methylation across regions. For more information, look at the methylation analysis video tutorial. Here we have mouse samples, 3 mouse samples across 2 groups that were compared, and we want to view their base level methylation rates using IGV. That file is downloaded already and is outputted as a VCF file. You always want to keep its concomitant.tbi file, which is the index file. So we will upload that file to IGV. So now we have the file uploaded for this mouse genome 3 sample comparison. We have the induced group and the control group. So you can zoom in to a chromosome level to see fine detail instances of cytosines that were measured by these samples are measured by RRBS. So you can zoom in for the cytosines that were assayed. Here, let me expand this so you can get a look at this. You have, for this genomic cytosine, we have output on for each sample here, for the plus strand and then the minus strand, induced sample replicate 1 on the positive strand. This genomic cytosine had a methylation rate of 100, as you can see under sample attributes, MR equals 100, and this is represented in red. So across all samples, you have relatively high methylation rates. Here, 81% methylation, 100, 100, 100. And see, here's an example of low methylation. So this is cyan, 0% methylation at these sites. So you can visualize the methylation rate for different genomic positions. Optionally, you can examine summaries of methylation over specific genomic regions. For these samples, we looked at the average methylation across CpG islands. The output of that file is an IGV format file that summarizes the results. So we upload this file to IGV, and here on the left we see that each track shows MR methylation rate for this sample with this code, induced replicate 1. So each subsequent track shows the methylation rates for those samples, and then methylation rate average for the induced group and the methylation rate average for the control group. We also see other tracks, like for example the number of sites that go into that methylation calculation, but we won't look at those now. So here in IGV, I changed the format of these methylation rate tracks to heat maps. And then I want to define the heat map range. Here we'll use 100 set heat map scale, and the maximum is 100. You can also optionally change the color, visualization color. So here I want to have high methylation be red, say low methylation be cyan to mimic our other scale. Let's do that. Okay, something like that. So now you see a different genomic intervals. Zoom in to a particular one. You can look at one CpG island, for example. Okay, so from here to here, let's see, is one CpG island. In this induced sample, the average methylation for all the Cs at this island was 100%. And now below it, we have our original VCF for the base level region. So we see that this estimate is made up of these several sites. And you see here induced, induced, induced. Those all show high methylation. So that goes into the average of this CpG island. So you can see using GobyWeb output, you can visualize methylation rates on the genome, whether at base level resolution or at genomic window intervals. This concludes our tutorial on GobyWeb and IGV. For more information, please visit our website at gobyweb.companionlab.org.